My name is Carla Costa, and I work in the Environmental Health Department in the National Institute of Health in Portugal. And I would like to thank the opportunity to the organization to present here some of the work that I have been developing in the past few years. So we all know that environment is responsible for the development of different diseases, and for many years, we have been only focusing on the external environment. And more recently, the concept of exposome. And I won't get into much details here because I know that a later talk will um, explore a little bit more that concept. But the truth is that we now know that we not only need to look at external environment, but also at internal environment. That was somehow a little bit what we were looking before when we talked about susceptibility of the individual. So this is related to some unique characteristics of the individuals. And in fact, this concept of exposome has brought some new challenges to environmental epidemiology. And one of these challenges is the need to consider simultaneous exposures. So what you see here is kind of a map for different exposures. And you can see that we will have personal environment. And in here, we will consider some of the exposures that we can somehow control. So we can control the way we cook, or the chemicals that we buy to clean our household, or if we smoke or don't smoke. But then we are also integrated in a social environment. So it depends on society if we go to a restaurant and if we are exposed to tobacco smoke in that context, or if there is too much traffic. So there are in fact exposures that we can control, but there are others that we can't control. And we can actually even talk about the natural environment that we cannot control at all. And in any of those cases, we are exposed to very different chemicals, and you can see here some examples. And we are exposed to them simultaneously. So this means that whenever we make or we perform a study and we are looking only at one exposure, in fact, we are somehow leaving behind the effects of all other exposures, and we may be biasing our results. Another challenge of the concept of exposome is related with timing of exposure. So we do know that the susceptibility of individuals to different chemicals is different depending on the life stage at which they are. And one of the most important life stages for that matter is the fetal stage. And we know that different studies have been showing that whenever individuals are in utero, are exposed in utero to different chemicals, there are the possibility to find effects in health later on. So we have the immediate effects. So immediately we have different alterations in perinatal outcomes, for example, or we may have different alterations when in childhood or adolescence or even adulthood. And we are talking about neurodevelopment, cardiovascular disease, or social and behavioral problems. There are a different number of possible health problems. If I give you the example of tobacco smoke, and I will give you that because it is still the main preventable cause of death, we can see in the literature that tobacco smoke in utero has been associated with perinatal death. It has been associated with alterations in child neurodevelopment. It has been associated to alterations in DNA. And also with epigenetic alterations. So based on all this information, we decided to design a project that was with the aim of analyzing the impact of this exposure to tobacco smoke and focusing on genetic and epigenetic effects. But because of the challenge of simultaneous exposures, we decided to integrate also different kinds of exposures. So in addition to tobacco smoke, we were considering also air pollution, both indoor and outdoor, and other relevant chemicals, for example, PAHs, metals, bisphenol, phthalates, and brominated polyphenylines. So showing you here the design of the project, you will see that we had different work packages. And the first one was about population recruitment, questionnaire administration, sample collection and storage. And then we had three main work packages. That one was related with exposure assessment. So we wanted to analyze exposure to tobacco smoke using a questionnaire. And then we had a second task to analyze simultaneous exposures. Then we had another work package that was focusing on genetic effects. So we were talking about the use of comet assay to analyze both DNA strand breaks and FPG sensitive sites. And then we had the epigenetic analysis. So we had DNA methylation, it would be global DNA methylation, and also histone modification. And then the final work package would be about data analysis. So talking a little bit about the population recruitment, the study was carried out in Centro Hospitalar São João in Portugal. And it, of course, received an ethical approval. And we were able to recruit 833 pregnant, healthy women from May 2017 to August 2018. And this was during pregnancy, during the wait for a medical appointment around the 37th week of pregnancy. So all women provided an informed consent. They also received a leaflet, and they had a small conversation with one of our co-workers to explain, to ask all the questions or doubts about the study and the possible implications of their participation. And then if they accepted, they signed an informed consent. So after accepting to participate in this study, they were signalized by the hospital. And what happened is that at delivery, the hospital staff was responsible for the collection of different matrices. So we collected mother blood, mother's urine, umbilical cord blood, umbilical cord tissue, and placenta tissue. We were able to get out of those 833 recruited women, 593 paired samples. So this means that in this case, we have all types of samples. In addition to sample collection, we also had this face-to-face interview, as I was saying before, to provide information through a questionnaire. So we were asking information on socio-demographic and lifestyle variables, tobacco consumption, environmental tobacco smoke exposure, both before and during pregnancy. In this case of pregnant women, well, these are susceptible populations, as I was saying before, but they also are very vulnerable. And this is a time point at their lives in which it raises different ethical questions to approach women at this time point where they are so vulnerable to actually collect sometimes invasive samples. So the possibility to collect placenta tissue that is not very frequent for genotoxicity assessment is actually an opportunity to initiate the establishment of the use of non-invasive matrices in this type of studies. So in fact, placenta is often a discarded sample, so it would be easy to get the approval of women to have that sample. And in opposition to cord blood, that is very limited and valuable. And as you know, and also in Portugal, many women will do the private biobanking of cord blood. And even those that are not doing the private biobanking, there is a national biobank, public biobank. And so even in the case of our study, that was always a priority for the hospital, first to collect for the national biobank and then for the study. So the use of placenta could actually be a good opportunity to initiate the analysis of in utero, of the genotoxic effects analysis after in utero exposures. I would like to show you some of the results we already obtained, and here you will see the results about the questionnaire, about the questionnaire and the questions about tobacco exposure. So we have already published last year the results about self-reporting. So what are the results of the questionnaires? And as you can see, 27.6% of women report to be smokers in the period before pregnancy, and then only 14.6 of them report to be smokers at birth. So this means that there is a good amount of women that actually cease smoking during pregnancy. About ETS exposure, the numbers are not that different. You can see that before pregnancy it would be 57.4, and then at birth 49.8%. The paper was very focused on the importance of socioeconomic position and cessation. So we also, one of our main results was that both smoking and ETS exposure were associated with lower educational level. We are now finishing another paper about smoking status, but this time based on cotinine assessment. We do know, or at least it is widely reported in the literature, that women tend to underreport exposure to tobacco smoke during pregnancy. And so what we did in this case was that we compared the answers that we had in the questionnaires with a cotinine assessment and then we have our main findings are the establishment of different urinary cutoff values. So we establish a cutoff value to distinguish pregnant non-smokers from smokers, and then differentiate non-exposed from exposed to ETS among non-smokers. And then when we compare these results with the results we had in the questionnaire, we actually found a very good agreement. So we do not support the previous finding of underreporting. Regarding simultaneous exposure, as I was saying before, this was one of our aims in this project. So in addition to tobacco smoke, we also had the outdoor air pollution and indoor air pollution. And regarding indoor air pollution, you will also can find available two already published studies. And we also included the determination of different chemicals in different matrices, and that is going to be carried out in the University of São Paulo in Brazil. Looking now at the genetic effects evaluation, you can see that so we had different samples, as I was saying before. We had the placenta, the umbilical cord blood, and the mother's blood. And both in the case of umbilical cord blood and mother's blood, we did mononuclear cell isolation before preservation. And regarding placenta, we had to optimize this protocol. So as you know, the placenta includes a variety of different cells and even structures. It has the parenchyma villus, the membranes, the zone of insertion of the umbilical cord. So it was very important for us to establish a standardization of the collection. So we asked the medical staff to collect a section of the placenta that would be always similar, always in approximately the same place. And we asked them to cut a section, as you see in the image. It was in the fetal, from the fetal side to the maternal side. And then this section would be preserved in sodium chloride and kept in the fridge until transport and further processing. We had the we usually were collecting samples in the hospital twice a day. So once in the morning, early in the morning, and then later in the afternoon. So we were trying to keep these samples in the fridge for the smallest amount of time as possible. Then the next question we had was, how were we going to obtain a suspension of isolated cells? And we tried two different protocols. We tried the mechanical dissociation and also enzymatic isolation, and using trypsin and DNase. And what we saw was that the levels of DNA damage were actually lower if we use the mechanical dissociation. So this is what we decided to move forward with this protocol. So briefly, what we did was we always worked on a cold surface and kept all tubes and microtubes in ice while working in the bench. And from that section that we were obtaining from the hospital, we removed both membranes, and we only used the tissue that was below the membranes. And then we transferred those sections of tissue to a plate, and we added ice cold PBS supplemented with EDTA, and minced all the tissue with scissors. And we obtained a suspension, and that suspension was aspirated using a needle, a syringe and a needle. And after going through the needle, it was transferred to a microtube. That suspension was washed and centrifuged twice, and this was very important to us because in the initial protocol, we didn't have these steps, and we were seeing a lot of debris in the slides. So it was very important to add this washing and centrifuging steps. And then finally, we were adding cryopreservation solution. So we were freezing cells in FPS and 10% DMSO, and then freeze them at -80. We still don't have the results for this for the NeoGene project, but what I want to show you here is we participated a few years ago in a study that was being carried out in the University of Aveiro, and that was a very small study. It only included 53 women, so it's very small compared with the number that we have. But actually, this was the opportunity to establish the protocol, and it actually worked like a pilot study for us to understand if we can actually carry out this protocol in a real human biomonitoring study. And even though the results were not significant in terms of exposure to, in this case, they were studying mercury exposure and tobacco smoke exposure, and those the biomarkers of exposure were not significantly associated with the DNA damage. But as I was saying, this was very important to us to establish, to see somehow how were the values that we were obtaining in terms of DNA damage. And you can see, and that's what I want to show you here. And you can see that they mainly vary from 6% to 18%. So we can have a good dispersion of values, and this was really important to us. And this is it. I would like to thank all the team involved in the project, from the National Institute of Health and ISPUP, the medical and nursing staff of the Hospital São João, and study participants, of course, for being volunteers, to Professor Fernando Barbosa from University of São Paulo, and for project funding, of course. So thank you very much.
Carla, you said that you first did the cell isolation at one point and then freezing. Is it working in both ways? Have you tried the opposite?
Yes, we tried actually. Before doing this study, there was this. We are already trying to to work on placentas, and a group has contacted us to see if we could do the comet assay in samples that they already had preserved. And we tried to do that. So we tried the cell isolation in tissues that were already frozen. But the values of comet assay were really, really high. So we decided not to move forward that way. And this is why we decided first to isolate the cells and then to freeze them.
Freeze them. Okay. And you said that you were doing DNA strand breaks and FPG sensitive side. Have you tried also on the same samples DNA repair assay maybe?
We initially the project was only focused on DNA damage and epigenetics, but later on we got funding for a PhD student, and in her work program we have established DNA repair to actually see if we can get some new information, and it will help us with interpretation of results, of course. So I think it will be really nice to have both DNA damage and DNA repair.
Great. We have one question in the chat box. I will read it. Has this trial already been standardized for mouse placenta?
We only did it for human placenta, but it is possible that it can be used to use in mouse placenta. I think so. There are a few studies working with placentas and genotoxicity assessments. Very, very few. You will, if you look in the literature, you'll probably find 2 or 3. But actually, in vivo, using animals, it will be more frequent. But there are very few studies. A few years ago, we thought about doing a review on this matter, and then after looking a little bit more to what we had, we decided that we are still not in the stage of doing a review. We still need to do just a map. We need to understand first what is present in the literature and which are the gaps. And I think it will be very helpful for people that are working in this area to find some studies that will help them interpreting their results and seeing even at the technical points how they can do certain things. But there are very few data still in this area.